Electronic counters. This is another method under cell number. This will give you a direct count, and an example here we will discuss Coulter counter. The basic idea of Coulter counter is that the basic principle for any automated cell counter is that the cell suspension will be forced through a small orifice, and these orifice will be connected to an electrode. That means two electrodes, both positive and negative electrodes, and electricity will be passed, and whenever the cells pass through this orifice, there will be an electrical resistance developed, and these electrical resistance will be recorded, and that will be converted into a number. This is the basic idea. And usually larger organisms are easy to count, like protozoa, algae and other yeast cells, but bacteria also it is possible to count. When a bacteria passes through this orifice, the conductivity decreases and the resistance increases. So, because when these cells pass, these cells are poor conductors of electricity, and since they are poor conductors, there will be development of resistance to the current flow that will be recorded. And this image shows you a better idea how this machine works out. You can see the small orifice noted as aperture, and there will be electrodes, and through this small orifice the cells are forced to pass, and usually one cell passes through the orifice at a time. Okay, so and the resistance developed will be recorded by the machine. And usually particles of size around 0.4 micrometer to 1600 micrometer will be measured in this machine. And usually this suspension or the cells has to be dissolved in an electrolyte solution, and that's one of the disadvantage also, because if a particular substance is not dissolved in that electrolyte solution, will not be possible to count. That is one limitation. Another limitation of this method is that this gives only the total count. It never differentiate dead cells and living cells. And another limitation is that sometimes there is a possibility that a non-cell particle also will be counted, because if a non-cell particle, which is a non-conductor of electricity, when passes through the orifice, that also may be counted as a cell. So that will give you a false count. The fluorescent activated cell sorter, or the FACS in short, basically works under the principle of flow cytometry. In flow cytometer, usually this is an apparatus used for the characterization, and also we can use it for enumeration also, or cell sorting also. Definitely there will be fluorescent dyes like acridine orange or DAPI or cybergreen, etc. will be used. And as already mentioned in the beginning, this machine usually it is the advantage that this machine can differentiate live cells and dead cells. And in brief, I am not explaining the elaborated mechanism of flow cytometry, but giving you only a brief idea. The cell suspension usually prepared in a liquid sheath or in a liquid stream, and after that means already which are stained with a particular stain, or will be stained with different stains, and these cells are passed through a small channel, and this channel has a laser beam connected, and the movement of the cells through the channel is in such a way that only one cell passes at a time, just like already we seen in the electronic counters. So when the cells passes through the laser, across the laser beam, there will be either a scattering or a fluorescence emission based on the fluorescent dye used, and these scattering or the scattering of the light and also the fluorescence emission will be detected by the detectors set inside the machine. So the light, scattered light and also the fluorescent light will be detected by the machine, and this will be used to find out the intensity of the cell. Usually the intensity will be recorded, and also by providing an additional add-on to the machine, you can sort the cells based on their intensities. Okay, that means you can separate, you can sort out a particular stained cell into a separate container. So in that way you can sort it out, and also you will get count of different cells. Proportional count method is a technique depending on microscopy. Here what we do is a standard suspension of certain particles like plastic beads, and whose number is known, or the particles number is known, is usually mixed with a particular volume of cell suspension, usually equal volume, and these two are mixed together, and from this suspension a slide is prepared. That means just like we do for other staining, we prepare a slide and stained and observed, and the cell number, and we will count the cell number plus the particle number. The count of both cells and the particles will be taken, and from that value we can estimate the proportionate number of bacterial cells in that suspension. For example, if we got a number of 10 beads and 50 cells in the or that is the average number per field, and if the number of particles in 1 ml of standard suspension is 25,000, that's already we know that value because we added the particle into the solution. So we know the standard volume or standard the number of particles in this particular solution per ml, right? So if so, then the number of cells per ml of the suspension will be calculated as follows. That means 50 by 10 into 25,000 equal to 1,25,000. That will be the cells per ml. This is a comparatively simple way to count the bacterial cells in a liquid solution, and this will give you usually a total count again. Not there is no differentiation of viable count and total count, only the total count will be given. Another method is called breeds count. This is another method for the direct count of microorganisms, widely used for counting microorganisms in milk. Usually a known volume of sample, usually 0.01 ml, is spread over a slide. Then this is heat fixed, stained and observed under microscope. Then usually we examine certain area, and you can take average. The number will be counted from a particular certain fields, and an average will be taken. And the cells per square centimeter is calculated by determining the number of microscopic field per square centimeter. So we take the number and find out how many fields will be there in the slide, so that you can correlate the number. So how to calculate? The calculation is like you find out the area of the microscopic field first. That means by using the equation pi r square, and that means it's a circle. Area of the microscopic field will be by the equation of pi r square, definitely. So you will get that as 0.02. Okay, and area of this smear is, if it is 1 centimeter square, that you can fix it, whether 1 centimeter square or even less or higher. If it is 1 centimeter square, so the total number of microscopic field will in 1 centimeter square will be around 5,000, right? And we use a term called microscopic factor. This is nothing but the area of smear divided by area of the microscopic field into the volume of the sample. That is the microscopic factor. That will give you a number 50. Okay. So if after calculating all those values, we can go for the actual calculation of total cell number in 1 centimeter square area as microscopic factor into microscopic field. That means you will get 2,50,000 as the result. Okay, so this will be the pattern how you find out the cell number. So the total number of cells in 1 centimeter square area will be calculated as the total number of cells counted into microscopic field into microscopic factor into microscopic field. Okay. So the number of cells, for example, if it is x is the number of cells counted in the field, and so according to this equation you will get 2,50,000 into x. That will be the value of, or that will be the number of cells per 1 centimeter square area. Another method for getting the bacterial number is by membrane filtration followed by staining. Okay, so usually one of the main limitation is that only liquid samples could be used for this method. Usually the liquid samples are filtered by means of a cellulose filter or a membrane filter, and these organisms which are already trapped in the filter will be subjected, or the filter itself will be subjected for staining. And usually the staining we prefer fluorescent staining, usually with DAPI or some other fluorescent stains will be used, and observe under fluorescent microscope so that you will get the number directly. Alright, usually this method is preferred for samples where we expect a lesser number of organisms, like drinking water, or in that kind of samples where we expect very low density of cell number. In that situations, we can use membrane filtration and followed by staining. And otherwise, instead of using a fluorescent stain, you can directly go for electron microscope without this staining procedure also. From here we will get a direct count, but here also the count obtained is a total count, not viable count. Determination of the most probable number is also listed as an indirect method for getting the number of bacteria present in a sample. And usually this is used for the enumeration of coliform bacteria present in water samples. And the media we used is usually lactose broth, inoculated with a sample and incubated for usually 24 hours and observe for acid and gas production. The acid production is detected with the help of the pH indicator incorporated in the media, and the gas production is detected with the help of Durham's tube, which we kept in the tube in the inverted position. So any gas production will be detected with the help of this Durham's tube. Usually we can perform this experiment either as a set of three or 5 sets, or that means there will be 3 sets covering, or in each set there will be either 3 test tube or 5 test tube. Okay, so there will be either a total of 9 test tubes if we are using 3, or there will be 15 test tubes if we are using 5 test tubes. Okay, and usually the volume of samples added is 0.1 ml, 1 ml and 10 ml into this 3 sets. And after that we incubate and observe for color change and or for positive result, that means acid and gas production, and the number is counted. And that number, like for example you have two tubes out of the 3, if we are using 3 set, then out of the 3 test tube, 2 test tubes in the set where we inoculated 10 ml is positive. Then in the next set, that means where we have inoculated 1 ml, in that we have 1 test tube which is positive, and in the next 3 set where we have 0.1 ml inoculated, and in there also 1 test tube positive, then the value is 211, right? And from this value, or using this value, we can correlate the actual number of cells or the most probable number present in the sample. and this is obtained by using an MPN index, or MPN table is used for obtaining this particular number, right? Then the MPN table is a standardized table. From there you can obtain the MPN index.